Chapter 18, Segment 1, pages 573 through 580. Attention selects stimuli for processing. Attention, also known as selective attention, is the process by which we select or focus on one or more stimuli for enhanced processing and analysis. This can be sensory or internal thoughts. Arousal is an individual's global level of alertness. The selective quality of attention is what differs it from arousal. Hermann von Helmholtz demonstrated we can shift the focus of our visual field covertly. We can keep our eyes fixed on one location while studying some other area in our peripheral field. The cocktail party effect is the selective enhancement of attention in order to filter out distractors. For example, filtering out other sounds of a party while listening to one person speak. Cocktails actually exacerbate the problem of filtering because alcohol lowers sensitivity for speech-related frequencies, and thus reduces one's ability to distinguish their friend's speech from background noise. There are limits on attention. Dichotic presentation, the simultaneous delivery of different stimuli to the left and right ears. Shadowing. In a study, participants are asked to focus their attention in one ear and to repeat aloud the material being presented to that ear. Participants could accurately report what they were hearing in the ear they were focusing on, but they could only report a very little amount of what had been presented to the other ear. If shadowing is sufficiently difficult, then even an advance warning won't be helpful. When someone's name is presented to the unattended ear, it is only noticed about 1/3 of the time. Inattentional blindness is the failure to perceive non-attended stimuli that seems so obvious it seems impossible to miss. Divided attention tasks are a task in which a person is asked to process two or more simultaneous stimuli. These tasks confirm that attention is a limited resource that is very difficult to attend to more than one thing at a time, especially if the stimuli to be attended to are spatially separated. Attentional spotlight. The shifting of our limited selective attention around the environment to highlight stimuli for enhanced processing. Having a single attentional spotlight helps us focus cognitive resources and behavioral responses toward the most important things in the environment at any given moment while allowing us to ignore the extraneous information. One of the most important functions of attention is to act as a filter by blocking unimportant stimuli and directing cognitive resources only to the most crucial events which protect the brain from being overwhelmed. Attentional bottleneck. A filter that results from the limits intrinsic to our attentional processes, with the result that only the most important stimuli are selected for special processing. Early selection model of attention is the unattended information is filtered out immediately, at the level of initial sensory input. Late selection model is the filtration imposed by the processing bottleneck, and it occurs at a later point in time, filtering out stimuli only after substantial analysis has occurred. Perceptual load. The immediate processing challenge presented by a stimulus. When we focus on a complex stimulus that requires a lot of perceptual processing, no perceptual resources remain for us to use on competing unattended items. These studies indicate that attention strikes a balance between early and late selection according to the difficulty of the task. Attention is deployed in several different ways. Sustained attention tasks are utilized by Helmholtz. A task in which a single stimulus source or location must be held in the attentional spotlight for a specific period of time. Stimulus cueing is a testing technique in which a cue to the stimulus location is provided by the stimulus itself. Stimulus cueing has revealed two general categories of attention. We can decide where to direct our attention. Voluntary attention is also known as consciously or endogenously controlled attention, and this is the voluntary shifts of attention and that our conscious top-down process of directing attention toward a specific aspect of the environment in accordance with our interests and goals. Symbolic cueing task is the most common experimental procedure for studying voluntary attention that was originated by Posner. Participants stare at a fixed point in the center of a screen and must press a key as soon as the specific target stimulus. The response being measured is reaction time. Each trial is led by a hint as to where the stimulus will appear. Valid and invalid cues affect our reaction time, are averaged over many trials and compared with trials of neutral cues. Valid cues are a single arrowable point in the correct direction of a stimulus will appear. Invalid cues are an arrowable point in the opposite direction of where a stimulus will appear, Neutral cues is a cue that provides no hint about the target direction or location. Individuals quickly learn to utilize cues to predict stimulus location by shifting their attention in a cued direction without shifting their gaze in anticipation of target stimulus. Through the use of valid cues, invalid cues, and neutral cues, we know that directed attention helps us perform better, faster, and with increased sensitivity to cues. Box 18.1. Reaction time responses from input to output. Simple reaction time. Tests that have the participants make a single response in response to experimental stimuli. For example, the appearance of a target. Choice reaction time. Tests in which a person is presented with alternatives and must choose among them by pressing one or two or more buttons. Reaction times in a choice reaction time test where participants determine if two stimuli are the same or different averaged about 300 to 500 milliseconds, and the delay between the stimulus and response vary depending on the amount of neural processing required between input and output. Brain activity proceeds from primary visual cortex to the ventral visual identification pathway, to the prefrontal cortex, to the premotor and primary motor cortex, to spinal motor neurons, to finger muscles. It takes the sensory system somewhere in the inferior lobe approximately 110 milliseconds to recognize a stimulus, and about 35 milliseconds more for that information to reach the prefrontal cortex. This is where a decision about which response to choose must be made. Simple tests may be as low as about 30 milliseconds for a decision to be made, but for more complex stimuli, it may take longer. Then it takes approximately 75 milliseconds for the movement to be put into motion. This is the time elapsed between the point at which the signal from the prefrontal cortex arrives in the premotor cortex and the point at which the finger pushes the button. Some stimuli grab our attention. Reflexive attention is also known as exogenously controlled attention. This is the involuntary reorientation toward a sudden or important event. Considered to be a bottoms-up process because attention is being caught and controlled via sensory inputs from lower levels of the nervous system. Peripheral spatial cueing task measures the effects of reflexive shifts of attention on stimulus processing. A task-irrelevant sensory symbol is presented in the location where attention is to be drawn, and after a momentary delay, the target stimulus is presented either at the same location, which is a valid trial, or somewhere else, which is an invalid trial. Two important effects are observed. A valid cue captures attentional and enhances processing of subsequent stimuli in the target location, but this is only when the interval between the cue and target is brief, and the cue will draw attention toward the target location. After approximately 150 milliseconds, though, attention wanders away and processing enhancement is lost. When the interval between the cue and target is longer, approximately 200 milliseconds and onward, detection of the stimuli at the location where the cue occurred is increasingly impaired. Inhibition of return is the phenomenon in which detection of a stimuli at the former location of a cue is impaired for latencies of 200 milliseconds or more. This probably involved to prevent reflexively controlled attention from settling on unimportant stimuli for more than an instant.